Hi everyone, in this video I'm going to walk you through the steps that you need for Project 218. We have these lists of species for two different samples, and what we want to do is we want to be able to sort the list. So we can do that pretty quickly with a tool called dplyr. You can think of this literally like a set of pliers. It helps you reshape data. I'm going to run library dplyr and I can see, oh, there's no packages. That's because this is a new workspace. Our Studio Cloud's actually giving me a little warning, so I'm going to install it. And while that's installing, I'm going to upload the files that I need that have the species in it. So I'm going to upload adult species.csv. Since my example project had a child and an adult sample, and then I'm going to upload the child species.csv. And we can see that the installation is happening in the console. We want to wait until it's fully installed. You'll see the stop sign go away. And what we're looking for in this bottom left portion of the window is for that greater than sign to come back, which means we can then load the tool. And once we've loaded it, now the functions are available. So I want to make two data frames that will have my species data. I'm going to call the first one my child, read.csv. And I'm going to start typing the name of the file and then use tab to make sure that I get exactly the right name. We have a comma separated variable file that is separated by commas, so we don't need to specify the sep. We can actually click on this to look at it and see, does it have a header? And it does have a header. That's the default, to have header equal to true, so we don't need to set that. But we should, just to make sure that it reads in correctly, set call classes. And we've been doing this for the last few assignments. We want to tell R what type of data is in each column. The first column is character data. That's the name of the species. And the second column is numeric data. That's the count. When I run this line, I end up with a data frame. I can click on it just to look and see, okay, does that look like the right data? And it does. Everything's coming into those two columns, reading in nicely. And if I look at the type of data, I can see it read in correctly as a character and a numeric vector. Because that worked so well, I can copy the line for the second data frame. My adult, read in adult species. And again, I'm going to delete the whole file name, start to type the file name, and use tab to complete it. Run it. And now I'm ready to write some comments. This is load dplyr package, read in files. Now I can organize the data that's in my child to only be the highest number of counts. And I can do this with a really simple line of code using a function that is built into the dplyr package. So I'm going to type my child % greater than %, and then look for the top 5 rows. When I type that in, you can see I now have a printout of what the top species are and what each of those counts are. I can do the same thing for the adult data frame. And it will print to the console. In your assignment, you'll want to change this from 5 to 10 so that you can look at the top 10 species. And now each of these are things that I can look up in the gut feeling knowledge base. This tool has a list of all of the known species that are being observed in microbiome experiments and what health data is related to those species. So if I download the file, the gut feeling knowledge-based master list, I can then search it for the different species. So I can use Control F, look for this species, and it does not have this particular species. Let me make sure that's just not there with the space. I can see if there's any related species, and it does not have any related species. Can look for the top species. And I see that this species is identified. I can read about it by going over to the last row. And I see that this, if it's in a low amount, it's been reported in intestinal disorders. Now, if I go back to my data, I look and I see that my individual person has this as their top species. So this is a healthy gut. Having the presence of this organism that if it's at a low amount can cause intestinal problems means that this person, the child sample in my case, has a healthy microbiome. So this is what you'll do for each of your top 10 species. You can see the code is actually very short to get the top 5. And all you need to do to change it to get top 10 is change that number in the function.